Hey everyone, how's it going? In the past few videos, we've been exploring the process of what happens when messenger molecules or antagonists bind onto the receptors of a cell and produce responses inside the cell. But this is a more simplified, simplistic view of the process of messenger molecules binding to receptors. And this is because we're only considering one type of receptor site. In reality, there's two types of receptor sites. So in reality, you have your main receptor sites, which are known as your orthosteric sites, and you also have a secondary area for receptors, a secondary receptor site, which is called the allosteric sites. Now, receptors on your allosteric site are usually secondary or regulatory, whereas your orthosteric site you can think of as your main receptors. So different things can bind onto your orthosteric receptors and your allosteric receptors to produce various different responses. In this episode, we're going to take a closer look at orthosteric sites and allosteric sites, and we're going to look at the differences between them and the possibilities that can happen when these different sites get activated by different things coming and binding onto your orthosteric and allosteric sites. Let's get into it. So all this time, we've been talking about agonists binding to receptor sites and producing responses. So the site that we've been considering this whole time, even though we haven't explicitly mentioned this, is the orthosteric site. So as we said, the orthosteric site can be thought of as your main sites. This is the main site where agonists can come and bind onto in order to produce responses. That's what receptors are mainly for, to produce responses. So your body's endogenous natural messenger molecules can come and bind to your orthosteric receptors in order to produce responses. And as humans, we can try to mimic these endogenous messenger molecules by creating similar messenger molecules in a lab, so they're synthetic. And these are called exogenous messenger molecules. They come from outside the body instead of inside the body. And they can mimic endogenous messenger molecules coming and binding onto the same orthosteric site receptors to produce similar responses. And we also talked about inverse agonists, partial agonists, and antagonists. These are all things that can all come and bind onto your orthosteric sites in order to produce responses. Now, things would be nice and simple if this was the only type of receptor we had to worry about, but unfortunately, there's a second type of receptor we also have to worry about, and this is called your allosteric site. So unlike your orthosteric site, which can be thought of as your main site, allosteric sites, this should have an E, sites. Your allosteric sites, it can be thought of as your secondary or regulatory site. It kind of regulates and modulates the response that's happening inside your cell. So different things can come and bind onto your orthosteric site to produce responses, but then things can come and bind onto your allosteric site to modulate and regulate these responses. And this opens up a lot of new windows for possibilities, and it adds a whole new layer of complexity to cellular responses, because it's not just determined by your orthosteric site anymore, it's also determined by the regulation by things binding to your allosteric sites. So before we get into the many possibilities of allosteric modulation, let's talk about the importance of conformation of proteins. So for proteins, conformation basically means shape, physical structure, and this conformation is very important because the shape of proteins determines their function. So one protein shape can do one type of function, and another protein shape can support different functions. So let's say a protein has a specific shape, a specific conformation. A change in the shape or conformation of a protein can lead to a change in the functionings of that protein because different shapes can support different functions. Now this applies to receptors too, because receptors are types of proteins. So the shape or conformation of a receptor can do specific functions inside the cell. And if the conformation of the receptor were to change, then the responses that, what that receptor can do, what things it can interact with, and the responses that happen inside the cell, all of that can change because, like we said, shape equals function, and a change in shape equals a change in function. So when you have a receptor that's just chilling on its own, it's at a specific conformation. But then when something comes along and binds to your receptor, it can change the conformation or the shape of your receptor. And remember, change in shape equals change in function. So now that something comes and binds to it and it has a change in the shape of your receptor, now it can interact with different proteins in your cell, for example, and activate different transduction pathways and produce responses. So when the shape of your receptor protein changes, different things can happen inside the cell. And different messenger molecules that come and bind to the receptor can change the conformation in different ways, affecting response and functions in different ways. So that's why things that bind to receptors can have drastic effects on what goes on inside the cell by causing changes in the shape of these receptors, because this opens up a whole world of possibilities. So let's go back to orthosteric and allosteric sites. In addition to messenger molecules binding onto orthosteric sites, now messenger molecules can also bind onto allosteric sites. Now remember, your orthosteric site is called your main or active site because messenger molecules binding onto your orthosteric sites can often produce responses. On the other hand, when messenger molecules bind to allosteric sites, this alone often doesn't really produce a response because your allosteric sites are, remember, they're regulatory, they're secondary sites. They regulate the responses that's being produced from stuff binding to your active or main site. So your messenger molecule will bind to your orthosteric site and produce responses. And then when your messenger molecule binds to your allosteric site, it can affect the quality of these responses. It can modulate these responses. For example, it can affect the potency or the efficacy of the responses happening inside your cells. Alright, so now let's focus on a few key aspects of allosteric modulation. The first thing is that allosteric modulation, it permits orthosteric binding. So because your allosteric sites and your orthosteric sites are in different locations, they're completely distinct, this means that things like modulators that bind to your allosteric sites, they still, like when they come and bind, it still allows for things to bind to your orthosteric sites because these are two distinct sites. Now this is very different than what we saw before. When we only considered your orthosteric sites, when something like an antagonist comes along and sits here and blocks it, then an agonist cannot bind because there's something in the way. So within orthosteric sites, only one thing at a time can bind because they're both, fighting to bind to the same receptor. But on the other hand, now we have orthosteric sites and allosteric sites. So if a modulator comes and binds to an allosteric site, then something else can still come and bind to your orthosteric site because remember, these are two distinct receptor sites. It's not like one or the other can bind like you have when you only consider orthosteric sites. So modulators binding to allosteric sites can be positive or negative allosteric modulators. A positive modulator, as the name suggests, it enhances or potentiates responses. So this could be, let's say, an increase in potency or an increase in efficacy, or both. Allosteric modulators can also be negative modulators, and as the name suggests, these lower or antagonize responses. So for example, they may decrease potency or decrease efficacy, or both. So you have positive and negative allosteric modulators that can enhance or antagonize your cell's responses. Okay, so let's say this is the standard normal dose-response curve of an agonist binding onto orthosteric sites and producing a response. So no allosteric modulation is involved in this dose-response curve. So now let's say we add allosteric modulators. These will go and bind to allosteric sites and they can modify or modulate your response. So let's say we're adding positive allosteric modulators. Remember, positive modulators enhance your response. And this can happen in a couple of different ways. For example, positive allosteric modulators may increase the potency of your agonist's original response. So they can shift your dose response curve back like this, making it look something like this so that you're able to get the same response but with a lower concentration, therefore increasing potency. Or it may increase its efficacy, allowing for a higher maximal possible response. So it may shift it shift the final plateau upwards so that the final max possible response is higher than your original response. So there's an increase in efficacy. Or maybe you can do both, increasing both potency and efficacy. So you need less concentration to have the same response, therefore higher potency, and you're able to get a higher maximum possible response, so higher possible efficacy. It doesn't have to be limited to just one thing. Allosteric modulation is very variable. Lots of different things can happen at the same time, and it opens up a whole world of possibilities. But the general thing to know is that positive modulators enhance response. They kind of boost the original response that the agonist was originally able to produce on its own. So now let's say that the allosteric modulator that we added was not a positive modulator, but instead a negative modulator. Well, as we said before, this lowers or antagonizes the normal response that the agonist produces. So here, this is the response that your agonist alone produced by binding to orthosteric sites. And now when we add our negative modulator, it will antagonize this response. And this can happen a number of ways. For example, it might lower its efficacy or it might lower its potency. So looking at our dose-response curve, when we have our agonist alone, this is our standard curve. But then when we add our negative modulator, for example, let's say it lowers potency, then it can shift our curve over to the right. So now you need a higher concentration of drug in order to produce the same response, and therefore potency has been lowered. Or it may lower its efficacy. So the final plateau of the dose response curve may be lower, so that it has a lower maximum possible response and therefore has a lower efficacy. Or maybe a negative modulator can do both. It can decrease potency as well as decrease efficacy. But the general idea is that negative allosteric modulators lower the responses of your original agonist. Allosteric modulators can be even more complex than this. They may combine the effects of positive and negative modulators. For example, adding an allosteric modulator could change an original response curve to look maybe something like this. So as you can see here, this curve has an increased efficacy, but it has a decreased potency. It has a higher maximum response, but it also requires more concentration to get the same level of response. Therefore, it's acting as both a positive modulator, but at the same time as a negative modulator. So there's a lot of room for complexity with allosteric modulators. So pharmaceutical companies, they can design drugs that act as allosteric modulators that go and bind to allosteric sites, and they can regulate the responses produced by agonists binding to orthosteric sites. They can increase responses and decrease responses in different ways, basically fine-tuning these responses to get exactly the responses that are desired. And this can help pharmaceutical companies design drugs that can produce very specific responses in the body to help it, maybe fight diseases or infections or relieve the body of certain conditions effectively. So you may now be wondering, how can an allosteric modulator binding to an allosteric site, how does it have all these effects? How is it able to impact the responses and effects produced by an agonist binding to your orthosteric sites? Well, it kind of goes back to the importance of protein conformations that we mentioned before. When modulators bind onto allosteric sites, what can happen is that the conformation of your orthosteric receptors can change. And this can change the way that agonists interact with your receptors, because if your receptors have a different shape, then maybe your agonists will be able to have a better fit onto receptors, or maybe they'll have a worse fit onto receptors. The way that the agonist is able to bind to the receptor is now going to change because the shape of the receptor has changed. And this can affect responses in many ways. So let's look at a really simplified example of this. Let's say you have your orthosteric receptor, which has a circular shape like this, and you have your agonist A with a square-like shape like this. And your agonist A comes and binds onto your orthosteric receptor, but because they have different shapes, it can't really get a perfect fit. But then, remember, This is a very oversimplified explanation. In reality, protein conformations are much more complicated than just, a circular or a square shape. There's a lot more that's involved, but this is just to, get the idea across. And then your allosteric modulator can come and bind to your secondary allosteric sites. And then when it comes and binds, remember we said that this can change the conformation of your orthosteric receptor. It can change its structure. And what can happen is it can change its structure to increase receptiveness to your agonist. So as you can see in your diagram, the binding of your allosteric modulator to your allosteric site has changed the conformation of your receptor from a circular shape into a square shape. And the square shape now has a better fit with your square-shaped agonist. So it has a better fit, it has increased receptiveness. And this increased receptiveness may potentially enhance response. So it might increase potency or increase efficacy. Now again, this is a very, very oversimplified explanation. While it's true that allosteric modulators do change the conformation of your receptors to either increase or lower responses, it's a lot more complicated than just, a better fit. And just because a receptor has increased receptiveness to an agonist doesn't necessarily mean it's going to increase the efficacy or it's going to increase the potency. This is just a possibility, just to get the idea across. But in reality, a lot more complicated. But you understand that allosteric modulators bind to allosteric sites, change the conformation of receptors to increase or decrease receptiveness, and in doing so, this can increase and decrease response. So it's important to remember that the opposite can also happen. Allosteric modulators can also be negative modulators. And when they bind to allosteric sites, they can change the conformation of your orthosteric site to have decreased receptiveness and therefore decreased response. So similar example as before, except now your agonist A and your orthosteric receptor, they have a much more similar square shape, which allows for a good fit. But then your allosteric modulator comes along and binds to your allosteric site, which causes a change in your orthosteric receptor to go from a square shape to a more circular shape. And now there's a worse fit between your agonist and your receptor. So there's decreased receptiveness, and this in turn can maybe, lower potency or efficacy or both. And again, just like last example, remember this is very, very oversimplified. Protein conformations are much more, they're much more in depth than just a square shape or a circle shape. And the way that allosteric modulators change these conformations and increase or decrease responses is a lot more complicated. But this is just to, help get the idea across. So by affecting the conformation of orthosteric sites, allosteric sites can regulate the effects of agonists, either increasing it or decreasing it. Now there's an important thing to consider. On some cells in the body, they can have receptors onto which different agonists can come and bind to either produce similar responses or different responses. So for example, in cell number 1 here, you have an orthosteric receptor and you have agonists A and B, which can come and bind to your orthosteric receptor and produce similar responses. But then over here on cell number 2, you have an orthosteric receptor and you have two different agonists, C and D, and they can both come and bind to your orthosteric receptor, but they are linked to different responses. So agonist C is linked to one response and agonist D is linked to different responses. So like we said, different agonists can bind to a same receptor on a cell, and they can either both produce similar responses as each other, or they can produce completely different response pathways. So as you can see, with agonists interacting with different receptors on different cells, things can get very complicated very fast. And allosteric modulation can complicate this even further by having different impacts on the different responses produced by your different agonists. So for example, let's say our orthosteric site receptor is circular, and we have two agonists, A and B, that can come and bind to our orthosteric receptor and activate different response pathways leading in different responses. But as you can see, agonist A has a square shape and agonist B has a triangle shape. So neither of them fit that well into your circular shape receptor. They're able to bind and produce response, but just not crazy strong. But then you add an allosteric modulator, which comes and binds to your allosteric site, and it changes the conformation of your orthosteric sites, so now it has a better fit and increased receptiveness to your agonist A. As you can see, the conformation of your orthosteric receptor has changed from circular to square-shaped. So now it has a better fit and better receptiveness to your agonist A over your agonist B. So therefore, your agonist A's response is likely to increase in maybe efficacy or potency. See, I've increased the arrow size and I've highlighted it to show that this response may experience enhancement, while agonist B's response doesn't really have a similar benefit because, it's still a different shape, so it might still have difficulty binding to this receptor. So the addition of the allosteric modulator doesn't really affect or change agonist B's responses, but it has a benefit for agonist A's responses. So as you can see, an allosteric modulator, it can be a positive or a negative modulator for one agonist while not having the same effects for another agonist. And as I've said again and again, the whole concept of, these different shapes of receptors and agonists is very oversimplified just to get the idea across. So one last thing to consider, another important thing to think about is affinity. We've talked about affinity in previous episodes, and it's basically the attraction between an agonist and a receptor. It's the ability for an agonist to bind and the ability for it to stick onto the receptor once it's bound. So when you add an allosteric modulator and it changes the conformation of your orthosteric sites, not only can it potentiate or enhance the responses of one of your agonists, maybe increasing efficacy or potency, but it may even increase affinity to one of your receptors over the other. So for example, in our case here, we're going, the allosteric modulator is binding and it's changing the orthosteric receptor's conformation from a circular to a more square-like conformation, resulting in better reception with your agonist A. This might actually increase affinity to agonist A. So there might be more attraction, more affinity compared to agonist B, which has less attraction and less affinity. So this means that agonist A is more likely to bind and stay onto your receptor as opposed to agonist B. And therefore, the response that is caused by agonist A may be more likely to take place. Because logically, if a receptor has greater affinity to one agonist, then this agonist is more likely to bind, more likely to stay bound, and therefore its response is more likely to take place. So then this response may be more likely to occur in the cell as opposed to our second response, which is caused by agonist B and has a lower affinity. So not only can the change in the conformation of your receptor, not only can it potentiate one agonist response while not impacting the other, but it may actually bias responses to make one agonist response more likely compared to another agonist response. It might favor the likelihood of one transduction pathway and response over another transduction pathway and response. So before we go out and close out, let's review what we've learned today. So cells have orthosteric sites and allosteric sites. Orthosteric sites are like your main or active sites. This is where agonists or ligands, endogenous or exogenous, can come and bind to, and they can directly produce cellular responses, hence the name active sites. Or allosteric sites, on the other hand, are more so secondary or regulatory sites, where allosteric modulators can come and bind onto it in order to regulate or modify the responses produced by your agonists that are binding to your orthosteric receptors. So they can maybe alter the potency or the efficacy of the response produced by your agonist that binds to your orthosteric sites. So you have positive and negative allosteric modulators. Positive modulators potentiate responses, meaning they can enhance responses such as increasing potency and efficacy. And now negative modulators can lower responses. For example, they can lower potency or lower efficacy. Now, when an allosteric modulator binds to your allosteric sites, it still permits things to bind onto your orthosteric site, because your orthosteric site and your allosteric sites are two completely separate sites. It's not like your agonists and modulators are binding to compete onto the same thing. Sorry, it's not like they're competing to bind onto the same thing. But when your allosteric modulator binds to your allosteric site, it can change the conformation of your orthosteric site receptor proteins to increase or decrease receptiveness to various agonists that may be trying to bind onto these receptors. So basically, your allosteric modulator can bind onto your allosteric sites, changing this orthosteric site conformation, and that in turn can alter orthosteric site and agonist interactions. So once the allosteric modulator binds to the allosteric site and changes the conformation of the orthosteric receptor, this new conformation may be better suited to one agonist, but less suited to another agonist. So it may potentially increase the responses or potentiate the responses of one agonist while not having such effects, or maybe even having opposite effects and antagonizing the effects of your second agonist. So maybe for agonist A, it can increase potency and efficacy while not really having such an effect, or maybe even lowering potency or efficacy for agonist B. There's all sorts of possibilities. And finally, as different agonists may be linked to different transduction pathways, so this is response 1, and then agonist B can be linked to a different transduction pathway, which leads to response 2. Allosteric modulation may favor some transduction pathways over others, because if this new conformation is better suited to agonist A and therefore there's a higher affinity between agonist A and the receptor, then it may be more likely that response 1 may take place over response 2, and so this response, there may be a bias where response 1 is favored over response 2 and is more likely to occur. And that wraps up this video on orthosteric and allosteric sites and allosteric modulation. Thank you guys so much for watching. I hope you enjoyed and found it informative. If you did, please consider liking and subscribing. And if you have any feedback or comments or anything you'd like me to go over again in more detail or explain separately, please leave a comment down below. I'd really appreciate it. In the next video, we're going to learn about different types of antagonism. So pharmacological, physiological antagonism, insurmountable, insurmountable, competitive and non-competitive. Antagonism is a whole new world, and we're going to break it down in the next episode. So until then, take care. I'll see you guys next time, and thanks again for watching.